first is just showing the difference between the typical vascular image that most people know on the left, showing the heart and the large arteries taking blood to the organs and the veins taking the blood back from the organs to the heart. And if you zoom in with the microscope, you'll see that way more microvessels can be found, and the majority of all the contact between blood and the tissue cells is in the microvasculature. And then the surface area of endothelial surface, communicating between the blood compartment and the tissues can be up to a few thousand square meters total, of which about 99% of all is in the microvasculature, and only about 2 square meters in the large arteries and veins. So almost all contact between the blood and the tissues is in the microvasculature, on the microvasculature endothelium. And when we started looking in more detail with microscopes in how those capillaries look like and how blood is flowing through those capillaries, these are one of the first images we got in the 1970s, 1980s, looking with microscopes in living muscle tissues. And we realized if you look at the individual red cells, how they are flowing through capillaries, you can appreciate that there's quite a big distance between individual red blood cells in these capillaries. And when people were trying, so they would freeze this frame, take a picture, and then count the number of red cells in the capillary, and they would know the volume of one individual red cell, and they would try to estimate the volume of the capillary by measuring the anatomical diameter defined by the endothelium. And if you divide red cell volume in this way by capillary volume, then the volume fractions of red cells, or tube hematocrit, were as low as 5%, 5 to 10%, which is obviously much lower than the volume fraction of red cells if you take a blood sample from a large vessel and you spin it down in a centrifuge, and you find 50% red cell volume of the total blood volume. So there was this big discussion why red cell content is so low. Is the blood diluted at the microvasculature, or maybe something else inside these microvessels that limits the volume available for red cell flow and plasma flow? And to make a long story short, the latter turns to be true. So people were able to take blood samples out of individual capillaries, and the blood is not diluted in the capillaries. So if you take a blood from the capillary and put it in a centrifuge, you back your regular 40 to 50% red cell volume fraction. So something else was in there. And then the first clue that we got that there's really a big glycocalyx occupying a lot of space is when I tried to image a glycocalyx or the protein stuck to glycocalyx by labeling plasma proteins with fluorescent dyes. So what I would typically do, I would start on top with a trans-illuminated image of a capillary, like in the movie, and then would see red cells with big spacings. And then in the dotted area, I would turn on a bright fluorescence light source to do fluorescence microscopy to see the fluorescently labeled proteins, see if anything would stick to the wall. And I tried to do that for 2 years and couldn't see anything sticking to the wall, until I realized that when I switched back to transillumination, even after 1 minute of light exposure in the dotted area, that exactly within that dotted area, suddenly the spacings between the red cells were much, much smaller. And the red cells would actually widen, and the flow velocity of the red cells would drop by about 50%. So just proximal to this dotted area, they would flow really fast, big spacings. They hit the exposed area, the cells would widen, get very close, and flow very slowly. And then when they leave the area that was exposed to the bright light, then suddenly they would speed up again, and the distances between the red cells would increase. So definitely we didn't change anything in the blood content flowing through the capillary, but something that we did in this dotted area caused the red cell content or the tube hematocrit to double or triple, even within a minute. So if glycocalyx was part of that low red cell content, by turning on the bright light, we probably had killed glycocalyx. So then I went back and looked in more detail how those red cells behave and the plasma behave in that dotted area before and after the glycocalyx damage. And then we got these kinds of images. So on the left-hand side is just at the beginning of the experiment. In the dotted area segment, you see only 2 red blood cells in this capillary segment. And then we realized if you look more closely, there is a significant gap between where the red cells are flowing in the center and the luminal surface of the endothelium. And also, if we look at the width of the plasma column, that we labeled large proteins like albumin with the fluorescent dye, and then measured the width of the flowing plasma column, that also that width of the plasma column is significantly smaller than the inner anatomical diameter. And on the right-hand side is the same capillary segment 1 minute later after turning on the bright fluorescence light for a minute. And then we realized that the red cell content doubled in that 1 minute. Let's see, 4 red blood cells instead of two. Now the red cells appear to be touching the endothelium, we're getting very close to the endothelium, while the anatomical diameter didn't change. But also the width of the flowing plasma column had significantly widened and now fills up the complete anatomical diameter of the capillary. So just by turning on the bright light, and now we know that the dyes that we were using, the fluorochrome and fluorescein, one of the most aggressive oxygen radical producing dye, if you excite it, that those oxygen radicals that we produce by turning on the bright light and exciting the fluorescein, well, quickly damage glycocalyx and break it down in such a way that a lot of more volume inside the capillary now becomes available for blood flow. So the red cells widen, the plasma between the red cells also widens, so the distance between red cells become much shorter, and it gets very close to the endothelium. So that was an important observation at that time. And then before we had the Glycocheck system, one way of assessing glycocalyx dimensions was then, by measuring the gap between the red cells and the endothelium, but also comparing the width of the plasma column before and after damage. So you can actually subtract the narrow column from the wider column, or similarly, inject two different molecular weight dyes, a high molecular weight that would distribute only in the center, and a low molecular weight that would easily diffuse into glycocalyx. Then if you subtract the narrow width from the wider width, you get another way of sort of positively staining glycocalyx. And that's how these images look like. So this is the two fluorescence images subtracted from each other. And you see some positive staining of a low molecular weight dye diffusing into glycocalyx. And so got more appreciation of the potential size and dimension of this glycocalyx where red cells and large proteins do not have access to. And just for to be on the same page again, if this is a really small capillary of about 5 microns in diameter, and you have, now let's say about a micron thick glycocalyx on either side of the vessel lumen, in terms of volume, which goes by the square of the diameter, so the anatomical diameter volume would be 5 square or 25, and then the functional blood perfused lumen would only be 3. 3 squares 9. So only 9 out of 25 would be blood perfused, and 16 out of 25, so 2/3 of the volume would be occupied by glycocalyx. Also now explaining why there's so few red cells in there when glycocalyx occupies more than 2/3 of the total volume. Now, using these techniques initially, by injecting small plasma tracers like small dextran molecules in the bloodstream and let it dilute into glycocalyx, gave us our first method to measure in humans whole body glycocalyx volume by a tracer dilution method. So we injected the dye, we let it circulate for a few minutes, and then measure the dilution of the dye that enters glycocalyx, and then comparing that with the blood volume that can be assessed by labeling red blood cells that do not have access to glycocalyx. So again, that difference gives you an estimate of whole body glycocalyx volume. And those first estimates are shown here. So these are 3 different kinds of experiments that we used healthy Amsterdam students, at least before we started challenging them. So on the lower left, we asked students to smoke 5 cigarettes in an hour, and then did the whole body glycocalyx volume assessment with the tracer dilution technique. And it was a significant drop after the hour of being exposed to cigarette smoke. Another thing that you might appreciate is if you look at the total volume of whole body glycocalyx volume that we assess in this way, there's a significant volumes of a liter or more, which also makes sense if you think about total body surface area of the endothelium, which is now maybe 1,000 or 2,000 square meters. if you multiply that with a 1 micron thickness, then that's the sort of volumes that you would get, like a liter or 2 liters of volume stored in the microvasculature predominantly, compared to the 5 liters of blood that are circulating. And out of those 5 liters that's circulating, we know that most of it is venous. It's about a liter in the arterial system, and maybe a liter or a little bit less in the microvasculature. But again, if we have that ratio of 2/3 volume occupied by glycocalyx and only 1/3 of volume accessible to blood flow, so if you would have 1 liter of blood in the microvasculature, that also means you have about 1 or 2 liters of glycocalyx volume total stored in the microvascular compartment. So that would be consistent with these early estimates of total body volume. Now, in
And
addition to the sure.
Hans, can I ask a question? I'm actually surprised how quickly you see this change. So in a span of I guess about an hour or so, or maybe you said 5 hours, how 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 acute of changes can you measure?
Well, it can be very acute. So, no, it's very reversible also. So that's a good thing. We
Yeah,
didn't
yeah,
destroy the health of these students.
right.
But also, we know very little about the mechanical behavior of glycocalyx. So under resting conditions, under low metabolic exposure, glycocalyx behaves like a solid gel that excludes fluids and excludes cell permeation. But there are certain triggers, like pH changes or ionic strength changes or pathogenic stimuli, like toxins from smoke or hyperglycemia or hyperinsulinemia or inflammation by exposure to endotoxin that can really easily make glycocalyx very, very permeable and give almost instant access to plasma volume and red blood cells. So that was one of the triggers also in the past that the people started trying to understand is not only I showed you that video, the red cell content is very low under resting conditions, but then if you um, activate the muscle and start the muscle contracting, or you add on top of that some vasodilators like adenosine, then almost instantly that whole volume that first was occupied by glycocalyx now becomes available for blood flow. So capillary blood content or total capillary blood volume can increase threefold almost instantly. And that's okay if you do it locally, but if you do that on a whole body level, all your organs at the same time, yeah, then you can understand you have a volume mismatch because now suddenly you have a liter or more additional volume that becomes available for blood, and you
Uh-huh,
don't have an extra liter of blood. So what we did see similarly, like on the top experiment, we were allowed at that time to inject endotoxin in those healthy Amsterdam students.
uh-huh,
So there was an almost instant additional volume in the microvasculature that became available for red cell and blood filling. And then you can understand it has a huge impact on their total blood pressure that went really
yeah,
significantly down. Heart rates went up. They got a little fever. And so that was mimicking almost like a septic shock situation.
yeah,
So we think that
interesting.
these volume mismatch, and actually similarly we see something also happen in pregnancy, for instance. So pregnancy really makes glycocalyx hyperpermeable. There's a lot of additional vascular volume that becomes available. And in some women, that challenge is more pronounced, and glycocalyx gets more permeable, more damage, and they're also more vulnerable for getting all kinds of issues with blood clotting, nitric oxide production that's impaired, leakage of proteins and fluids from the vasculature. So yeah, it can be, if you have a systemic, acute, big enough challenge, it can be instant and
Yeah.
very, very pronounced.
Okay, thank you.
All right, so then these experiments are fun to do, but they were laborious. That took hours for one patient to be measured. And then also the tracers that we use, the small molecular weight dextrans, we know they can induce anaphylactic shock in a small percentage of people. So not scalable. So we moved on back to our original observations that if you look at these capillaries with transillumination, we can just monitor the position of the red cells. And again, you realize, you appreciate there's a big gap between the red cell, where the red cells are flowing and where the endothelium is. So we now know that this space between the red cells and the endothelium is not just filled with flowing fluid. there's hardly any fluid moving into that space. It's all filled with mechanically stable glycocalyx. And then to further illustrate that, we worked hard on optimizing electron microscopic staining techniques and fixation techniques to get a positive stain of glycocalyx that you probably have seen. So it took a while to get these images. So it was a few years later, after we had the light dye treatment technique and the tracer dilution technique, we were able to positively stain the glycocalyx polysaccharides with a dye called Alcium blue. And the reason it took so long is that these structures that are extending from the endothelium. So again, this is a cross section of a capillary. This is a myocardial tissue. This is tissue cells. This is the lumen. Here's the endothelium, and these are the glycocalyx bushes extending into the lumen. On the right-hand side, much higher magnification. So on top is where the red cells are flowing. This is the endothelium, which is very thin in these microvessels, like a tenth or 2/10 of a micron. And then we have these extensive polysaccharides that we, for the first time, could show using the dye Alcium blue. And the reason it was so difficult to show is that these polysaccharides only occupy a very tiny fraction of the total volume of glycocalyx. It's maybe 1% or even less than 1% of the total volume is filled with these polysaccharides. And the rest is proteins that bind to the negative charges on glycocalyx, and then a lot of water that's pulled in to the high protein concentration that's inside glycocalyx. And then when you do your conventional preparation of your tissue samples for electron microscopy, you have to dehydrate your tissue samples. And you can imagine if you take away the water inside this matrix, you don't stabilize the structures before you do, all these polysaccharides collapse on the endothelial surface and almost disappear into like a nanometer scale kind of coating that people have been ignoring. So it took a long time to develop a protocol by staining these structures with Alcium blue, which is a cationic dye that binds to the negative charges on the polysaccharides. And then staining it also encapsulated the polysaccharides with the dye molecules and stabilized the structures at the same time. And then if you dehydrate, the structures keep standing and are showing up on the pictures. But it has quite a few
Yeah, those
challenges.
are really beautiful images.
Yeah, it took a long time
Yeah.
to get those. The poor Bernard Vandenberg, the first author on this paper, it took him about 3 years or 4 years to optimize the method to get these images. But more than 20 years ago, I guess if you try to do that now, you would, pretty much destroy the academic career of your PhD student or postdoc if it takes so long to publish their first paper. Huh?
Yeah, all my students are looking at me like, don't even think about it.
No, this is no, it's no, you don't want to do that to your students. It's still now, even knowing the recipe and trying to get it done. Because the other
Yeah.
problem is these structures are embedded in a gel under in vivo conditions, so which allows them to withstand shear forces, and they're robust and stable. But as soon as you start staining and you are successful in staining these structures, then they become very fragile. So they lose the gel environment. And then you need to keep perfusing them because the dye forms micelles if you don't perfuse. But then you have to lower blood flows to like a tenth or almost 100th of normal physiological flow rates, because then the shear forces acting on these coated stabilized structures become too much, and they all break down and disappear anyway. So yeah, you don't want to go there. Just uh, be happy that they were there and be convinced there's a glycocalyx. So that really helped to show people there's something in that space and allowed us to do some more experiments to see what the functional implications are of having a glycocalyx or damaging glycocalyx by exposure to risk factors or different enzymes like heparinase or hyaluronidase. We're going to show a few examples, if that's okay with you, what happens in those experimental settings. So one of the areas that we were looking into is atherosclerosis, looking at large artery bifurcation areas at the sites where that are predisposed to develop atherosclerotic lesions that are highly permeable. And then we realized that these sites where the lesions start developing, where the cholesterol leaks into the vessel wall and inflammatory cells start migrating into the tissue, those sites have a much thinner glycocalyx, even before atherosclerosis starts on the baseline conditions, than the rest of the vasculature. So downstream, or even at the opposite side of the same area, with very thick glycocalyxes. Not only these sides, they are very, very thin. And then when we started looking at the flow profiles of blood over those bifurcations, so this is a simulation of the blood flow over carotid artery bifurcation. And you see, so the red color is forward motion and the blue color is backward motion of blood. So you realize it just downstream of the bifurcation here at the sinus region that the flow profiles are very complex. So the blood starts circulating, and there are areas, small areas of few endothelial cells here in the wall where the blood is not moving at all. So there's no forward motion of blood over the endothelium at these sites because of these complex flow profiles. And then we realized that the endothelium really needs to feel shear forces, net forward movement of blood over the surface of the endothelium to stimulate the endothelium to start producing glycocalyx. So these sites where there's very little motion at all or even stagnant blood, the endothelium doesn't feel any trigger to start producing glycocalyx. So glycocalyx is very thin at this very localized sites with these disturbed flow profiles. Yeah, we could simulate a little bit more, like in flow chambers like these. So these are little chambers in which we can culture endothelial cells and then close them up and look through a window to measure glycocalyx production. And then we can control the flow of blood over the endothelium. And again, confirming that if we do that at normal levels of flow, we have a normal level of glycocalyx production. And then proportional with no lower flow, even very low flow, then glycocalyx production stops and it's very minimal. So you have very thin glycocalyxes at sites of very low blood flow or shear forces. So one of the consequences is in terms of permeability. So this is a study in which we measured the leakage of cholesterol molecules over the endothelium. So the cholesterol was labeled with the green dye and injected in the vasculature and then measured with confocal microscopy where the dye went. So if you look at larger vessels like the common carotid with the thick glycocalyx, you see now this is about 5 or 10 minutes after injecting the cholesterol into the bloodstream. And on top in the black area is where the blood is flowing. And then the blue dots is the position of the endothelium. And you see that in healthy vessels with a thick glycocalyx, most if not all of the cholesterol gets stuck in glycocalyx and doesn't leak into the endothelium. And in contrast, a little bit more downstream in that area of disturbed blood flow and the very thin glycocalyx, those sites are highly permeable. And you see not only is there's the cholesterol there in the glycocalyx, but also within the endothelial cells leaking into the subendothelial compartment, accumulating in the vessel wall at these sites of very small glycocalyx that are hyperpermeable. And that's really the early trigger that starts the inflammatory process and ultimately can lead to atherosclerosis. Well, another consequence is in terms of nitric oxide production and sensing shear forces. So we now know that shear forces by itself, those forces are way too small to mechanically activate the endothelium and trigger any biochemical process. So they need an amplifier, and glycocalyx is that amplifier on the surface that bends forward and acts as a lever that amplifies those shear forces about 100-fold, and that's enough to cross the threshold for biomechanical activation of your endothelium. So if we look at healthy vessels again with the thick glycocalyx, and we can increase the blood flow through these healthy vessels, we see a nice and proportional increase in nitric oxide production, which is pretty much absent the moment that you remove glycocalyx. And in this case, we use enzymes like hyaluronidase to take away the hyaluronic acid and the rest of glycocalyx. So there's still big ranges in blood flow, but the endothelium is unable to sense it. And there's very little increase in nitric oxide production when blood flow increases. At the same time, the endothelial cells can still produce nitric oxide if they're stimulated by chemicals like acetylcholine. So on the right-hand side is the same vessels with and without glycocalyx stimulated with acetylcholine, and you see still normal levels of nitric oxide production by this endothelium. So the endothelium is still there, still functional, can still produce nitric oxide, but if you take away the glycocalyx, you lose that shear sensor that allows you to increase nitric oxide production when blood flow increases. Another consequence is on leukocyte adhesion. We know that leukocytes cannot stick to the endothelium when there's no whole body contact. So this was a study on small venules. So a little bit on the left-hand side, proximal to the field of view, we impaled the venule with a small glass micropipette and injecting small amounts of saline or saline with enzymes to degrade glycocalyx every few seconds into the vessel. So you see when we flush out the blood every few seconds, you see the slowly moving, transparent, bigger cells, which are leukocytes, and they are slowly rolling over the endothelium much slower than the blood flow. And that's the normal physiological rolling behavior of the leukocytes. And they can do that in the presence of a healthy glycocalyx because the rolling adhesion molecules are on the tips of the microvilli, the fingers that stick out of the leukocytes. So on the surface, you have these fingers sticking out of a micron or longer with the rolling adhesion molecules on the tips of those fingers. And those fingers can easily penetrate glycocalyx and scan the endothelial membrane and adhere to rolling adhesion molecules. So they can roll slowly and they can patrol and check whether it's an inflammatory site, but they cannot firmly adhere and stick to the endothelium because for that they need full body contact, which they cannot have if there's a glycocalyx in between the endothelium and the leukocytes. So we could test what happens with the sticking of the leukocytes when we damage glycocalyx, either with pathogenic stimuli like high levels of cholesterol or specific enzymes like heparinase. So these are two examples of the levels of adhering leukocytes on the vertical axis as a function of time. So the blue data is just control data, very low levels of leukocytes sticking to the endothelium as long as glycocalyx is intact. And on the left, at the dotted time, we injected very aggressive oxidative levels of LDL cholesterol. And you see within half an hour, the oxidized LDL cholesterol significantly damaged glycocalyx and a significant increase in the number of leukocytes sticking to the endothelium. Similarly, on the right, more specifically, when we microperfused together with the saline injections, enzymes like heparinase, as soon as you start degrading glycocalyx, there's a very significant increase in the stickiness of the endothelium, and leukocytes start sticking in much higher numbers. One more example of a functional consequence if you damage your glycocalyx. So this is back to the capillaries. You see on the left a healthy capillary with the thick glycocalyx. On the right, you see what happens if glycocalyx is compromised. And again, here in this experiment, use enzymes like hyaluronidase or heparinase. So the glycocalyx bushes are gone. And at the same time, you see all this white space now developing on the outside of the capillary, in between the outside of the capillary and the tissue cells, which is pericapillary edema, the leakage of a lot of fluid and proteins out of the capillaries accumulating on the pericapillary space, a lot of interstitial swelling, also compressing the capillaries, so they become a little bit smaller in diameter. At the same time, the endothelium becomes very sticky, so a lot of cells start sticking to the wall, causing a lot of capillaries to stop being perfused with blood flow and not become functional anymore. So this is another example of how big that effect can be. So this experiment, Cabrales et al. Measured the functional capillary density, so how many capillaries are perfused by blood. You see even within half an hour, if you systemically disrupt glycocalyx, they lose about 40 to 50% of their blood perfused capillaries. So a lot of the microvasculature shuts down as soon as you systemically disrupt glycocalyx, like you could do for like a systemic infection with endotoxin, for instance. So yeah, you lose a lot of your microvasculature.
Thanks for listening to The Future of Vascular Health. Healthy vessels, healthy future. If you're ready to take charge of your health, it's time to check out Revasca. This powerful supplement is designed to protect your glycocalyx, the microscopic layer that keeps your blood vessels healthy and strong. Revasca can help support your heart health, improve circulation, and even reduce high blood pressure. It's also great for boosting brain function, keeping your kidneys healthy, and preventing vascular complications before they start. Think of it as a way to keep your body running smoothly so you can live longer, feel sharper, and stay active. Don't wait. Take the first step toward better health today. Visit glycocalyx.com to learn more about Revasca and how it can help you protect your future.